Hello everyone. In this video we will see nanoparticle corona formation. What is nanoparticle corona formation? So at the end you are going to learn nanoparticle corona, types of nanoparticle corona, process of corona formation, biocorona, impact of nanoparticle corona formation, host cell response to nanoparticle corona formation, properties of nanoparticles responsible for corona formation. So nanoparticles are used in various applications due to nanoparticle features like longer circulation times, increased solubility, sustained delivery, protection from physical and chemical degradation, and controlled release of drugs. However, these nanomaterials under clinical research shows unsatisfactory results. It is because of lack of an accurate understanding of their in vivo performance at systemic, tissue, and cellular levels. So, nanoparticle corona is when nanoparticle comes in contact with biological fluid. Nanoparticle absorb biomolecules like proteins, cholesterol, triglycerides, and lipids, and it forms a corona. This is called as biocorona. So the layer which is formed on the nanoparticles, which is of biomolecules, is called as nanoparticle biocorona. So formation of the biocorona is a dynamic process. It is driven by minimization of nanoparticle high surface free energy, in which different molecules of the biological fluid compete for the nanoparticle available surface. And corona formation is continuous and competitive process through dynamic interaction of protein and other molecules to the nanoparticle surface. The composition changes over time by displacement of earlier absorbed proteins or by other proteins with stronger binding affinities until equilibrium is reached, and this effect is called as Broman effect. Types of nanoparticle corona. So protein, lipid, and lipoproteins forms the corona. So mainly proteins is the in the biocorona have been extensively studied, and understanding of lipid coronas remains poor. And about lipoproteins, these are complexes of apolipoproteins, phospholipids, triglycerides, and cholesterol. In several studies, proteomic analysis suggests an enrichment of apolipoproteins in the protein corona of nanomaterials. Hellstrand et al. Were the first to describe that lipoprotein complexes as a whole interacted with nanomaterials. Lipid adsorption was further described in carbon nanotubes after inhalation. Synergistic interaction between the prevailing lipids and specific proteins were observed. So, biocorona is responsible for physical and chemical modifications on the nanomaterial, such as size, aggregation, and surface properties. And aggregation confers it a biological identity different from the primary synthetic identity. So, for example, you administered gold nanoparticles in the body. So, after going to your body or circulation, will they be the same gold nanoparticles which you have administered? That is the question. Next is influence of biocorona on nanoparticles. So it influences nanoparticle circulation half-time, biodistribution and uptake by cells, host immune response, toxicity and oxidative stress. The adsorption of proteins is influenced specifically by nanoparticle properties, media, and exposure time. In nanoparticle properties, particularly size, hydrophobicity, charge, surface chemistry, these are affected, and protein source and concentration of media is also responsible or affected. So, this knowledge why we should learn about nanoparticle corona formation. because the knowledge on the corona formed on nanoparticle biocorona determines how host cell responds to nanoparticles, and it helps in understanding what role it might have in vivo studies at systemic level, tissue level, and cellular level. So the models are used to investigate corona formation are mostly mouse models, which are used in preclinical and toxicological experiments. These are very commonly used, and nanoparticle interactions with mouse serums are also people or researchers over the world are studying. So identification of protein composition of biological corona is done by mass spectroscopy, and changes in size and zeta potential were observed, as well as the protein composition of biological corona, which I have said it's observed with the use of mass spectroscopy. So thank you. And then this is the emerging field, and specifically researchers over the world has that basic question, why and nanoparticles are responsible for causing toxicity, particularly to immune toxicity and deposition in different organs, and yes, they are responsible for toxicity as well. So but why and how? So and because, so even if we take some measures like coating of nanoparticles, this also affects the formation of corona, and this is how which is responsible, because our body behaves or acts this against as a foreign material. So that's why maybe these things are responsible for toxicity. Thank you all.